Hello, my name is Robin Shuttleworth. I'm a postdoc at the University of Saskatchewan. In my presentation today, I will be talking about the general mass tissue transfer model for cryopreservation applications. Before I start, I just want to acknowledge my co-authors on this paper, Dr. Warner, Dr. Iroglu, Dr. Benson, and Dr. Higgins, without whom we wouldn't have done this work. So in order for us to perform successful cryopreservation, there's 4 different stages that we have to perfect. And we are going to be focusing on the first and the last stage, which involves the addition and removal of cryoprotective agents. Now, these stages come with a few challenges that we have to try and overcome. The first of which is chemical toxicity. Now, the CPAs are highly toxic to the biological sample, and they will cause irreparable damage to our cells or tissue. The second type of challenge that we have to try and overcome is this osmotic stress. And when we consider tissues in particular, there's 2 types of stress that we are concerned with. And the first of which is osmotic stress. And osmotic stress is the change in size, really, of the cell or the tissue that we have. So if we were to place our cell into a hyper or hypotonic solution, we would see shrinking or swelling, and we would see the same type of behavior with a tissue sample as well. The second stress that we have to concern with is elastic stress. And elastic stress describes pressure that arises from the change in volume of the tissue with respect to the solids portion of the tissue. Now, the solids portion of the tissue is any part of the tissue that can't actually leave the tissue. So this would be things such as the extracellular matrix or the cells within the tissue. So any change in the volume of these, and we will see either a shrinking or a swelling happening with the tissue size. So this type of modelling of mass transfer has been done quite successfully for single cells, and we want to take this to a tissue level. So what are the challenges that arise when we try and go from single cell to a tissue level? Well, there's 3 main things that we want to try and consider when we're moving from single cell to tissue. The first of these is we want to consider extracellular mass transfer within the tissue. So this would be the part of the tissue that isn't solids, it's not cells, but it's just this extracellular fluid that's floating around inside the tissue. And we want to focus on the mass transfer of this when we look into tissues. The second thing we have to consider is any tissue size changes. Just as a cell will change size, it will shrink or swell, a tissue will do the same. So we have to work out how we're going to predict the size change of the tissue from one time to the next time. The last thing we want to consider are fixed charges. Now, fixed charges are these positive ions that are captured within the fluid phase of the tissue. And they're captured here by these fixed negatively charged ions on the proteoglycans. And these make us need to set up a kind of uneven or unequal distribution of ions inside and outside of the tissue. And this unequal distribution of ions has implications for the mass transfer of all the substances within the tissue. So when we're moving from single cell to tissue, we want to consider these 3 different things. So we can then take a look at our approach to building this mass transfer tissue model. So we begin with a basic mixture theory model, as in the one developed by Abazari. And we begin with a simple mixture of water, CPA, salts, and solids within our tissue. And we assume the water, the CPA, and the salts can be exchanged with the bath solution. The second thing we consider when we tackle this problem is we want to use all this tissue-based phenomena that I just described, so this extracellular mass transfer, these tissue size changes, and of course fixed charges. And the final big kind of addition that we will make to this model is we will include the addition of cells and therefore cell membrane transport. So we will assume that we have cells within our extracellular fluid, and these cells can both gain and lose water and CPA. So that's going to be our extension to the current model that Abizari developed for articular cartilage. Articular cartilage has very low cell density, which was why cells were initially excluded from this model. However, we want to expand this model to be adaptable to any type of tissue, and that means we have to have cells included in this new tissue model. So this is how we kind of are taking our approach for this model, so we can then start to build an idea of what our model is going to look like. So our model schematic looks something like this. So we have our tissue on the left in pink, and we have our bath on the right in blue. And our bath is going to contain a solution of water, salts, and CPAs. And our tissue contains 3 different compartments. We have an extracellular fluid compartment, we have an intracellular fluid compartment, which is going to be all the fluids within the cells, and we have this intra and extracellular solids compartment also. Within the intra and extracellular solids, we're going to have things like the ECM, the inactive volume of the cells. This is going to be considered to be the solids part of the tissue. We can also see we have some dashed lines here, which signify to us where the exchange of fluids will occur. So we can exchange fluids with the bath through the extracellular fluid compartment. And then to exchange fluids with the cells, these can only be exchanged through the extracellular. So the extracellular and intracellular compartments will exchange fluids also. So to describe all of these interactions that's happening, we can build a system of equations. And we build a system of 6 equations. We have 1 for each component within our system. So you can see we have 3 equations here for extracellular components, or water, CPA, and salt. We have an equation for the solids. And then we have 2 equations for the intracellular components, 1 for water and 1 for CPA. Here, we don't have an equation for intracellular salt. And this is because we assume that the cell membrane is impermeable to salt. So the salt we start off with in the cell is the salt we will have throughout. Now we made a big addition to this model by adding in this cell membrane transport. So we have to illustrate this within these equations. And we do this with these final terms highlighted in red here. And for our cell membrane transport, we are using a simple 2-parameter model. The 2-parameter model describes the exchange of fluids of a single cell. And we assume in our system that our cells are going to act homogeneously, so all cells will act the same. So we will do our 2P model for one cell, and we will multiply it by the number of cells that we have in each of our individual spatial domains. So one thing I mentioned at the start was we were interested in this tissue pressure that we had in our model. And we describe this pressure by any change in volume fraction of the solids part of the tissue. And we can see that the tissue pressure is signified by this E here. And this just gives us any change in the volume fraction of solids. And what we can take from this is if we have an increased solids volume fraction, then we have a negative strain and the tissue is shrinking. And if we have a decreased solids volume fraction, this will give us a positive strain, and this will tell us that the tissue is swelling. So it's kind of the main parts of our model. There's lots of little bits in between, but I just wanted to show the main equations of the model. The substances themselves are driven by chemical potential gradients, but it's lots and lots of equations, so we'll just stick with this for now. So there's 2 more parts to this model that I have to describe before we can take a look at some results. The first of which is the initial conditions. So for the initial conditions, we assume that the tissue is equilibrated with an isotonic solution containing no CPA. So this means that we assume there's our intracellular CPA is equal to 0 at initial time. And we assume the salts, both intracellular and extracellular, are equal to one another. So we have no movement of any substances at the initial time. We can then move on to consider our boundary conditions for our tissue. So we have 2 boundaries in our tissue because we are working in a 1-dimensional system. On the left, at x equals 0, we have what we call an insulated boundary. In the previous model for articular cartilage, this was considered to be at the bone, but because we are working with any type of tissue, this is just called an insulated boundary. And at this boundary, we have no movement of any substances. So the chemical potential gradients are 0. On our moving boundary, so the boundary that is in contact with the bath, so x equals h of t, we assume that the tissue is in contact with a fully equilibrated bath solution that contains some CPA. Okay. So now that we kind of set up our entire model, we have our initial and our boundary conditions, we can then go ahead and run this model for some different tissues. So the first 2 tissues we ran this for is we ran for articular cartilage on the right, which we had previous work from Abazari et al. And then on the left, we also ran it for a pancreatic islet as well. And I have put on either side of these the differences in these 2 types of tissue. So there is a few differences between the 2, the first of which we have to work in a different geometry for these. The length scale is different. The islet has a higher cell and solids fraction compared to articular cartilage. However, it has a much lower fixed charge concentration. And this again is because this articular cartilage has a very, very high fixed charge concentration compared to almost any other tissue. And from these 2 results, we can kind of make a couple of different predictions. So on the top graphs, we have the CPA concentration. So the islet was immersed into a 1.5 molar concentration bath of CPA, and articular cartilage was immersed into a 6.5 molar DMSO bath. And we can gain some predictions about the CPA concentration over time within these kind of cells in our tissue. So we can look at both the surface cell, which would be the cell that's in contact with the bath, so closest to the bath, and also with the interior cell. And this interior cell is the cell that's closest to the insulated boundary. So we can predict the CPA concentration in both of these throughout the entire tissue, and this means that we can then also predict for CPA toxicity as well. Second type of predictions that we can make are on volume changes within the tissue. And again, we can look at this for the surface cell, the interior cell, and also the entire tissue itself as well. And from this change in volume, we can then calculate the strain or the stress that's placed over the entire tissue. And from these 2 predictions we can then, when we move forward with this model, try and minimize both the CPA toxicity and the stress that's placed on this tissue. Now, there is one main difference that I want to point out between these 2, and this is this difference of time and length scale. So because we have a much smaller length scale for our islet, because it's smaller, that then translates to a difference in the time scale as well. So once we had a look at a couple of different tissues like we have here, we then wanted to take a kind of look at the parameters that we were working with and do a kind of analysis and find out the relationship between a couple of the different parameters. And the 2 that we chose to take a look at were the stiffness of the tissue, so what's known as aggregate modulus, and also the fixed charge concentration within the tissue. So on the left, we have 2 graphs with a high stiffness, so this would be something like articular cartilage, and on the left, we have a tissue with a much lower stiffness. On the top, we have a comparison of CPA concentration over time throughout the tissue, and then on the bottom, we have a volume change prediction. And we can see when we look at the DMSO concentration, they're quite similar, not too many differences between the stiffness, either high or low, and also when we change the CPA, the fixed charge concentration, there's not a lot of change in the CPA concentration either. However, when we go to the graphs on the bottom row and we look at the high stiffness, so the high HA of our tissue, we can see that when we have also a high fixed charge concentration, we get a much larger volume change of the tissue compared to when we have this 20 or 2 concentration on our fixed charges. If we take a look at low stiffness of our tissue, again, we can see we kind of get the same sort of volume change happening, the same minimum volume change. However, when we have high fixed charges, we can see we get this kind of rebound of the volume. So our volume is increasing again. Whereas when we have a low fixed charge and a low stiffness of tissue, we don't really see any rebound. We see it kind of leveling off and equilibrating towards the end of this time frame that we have. So this was just kind of a look to see this relationship between stiffness of the tissue and also fixed charge concentration. So moving forward with this model, there's lots of different things we can do with this model to move it forward. However, we want to kind of look at moving this into a 2D environment, so that rather than the 1D model we have right now, we're going to place it into 2D, so we can look at the movement of substances in both the X and Y directions. We can then start to test out different and more compositions of the tissue in the bathing solutions, so we can get high and low cell fractions within these, and then we can also alter the CPA concentrations within the bath. We can also then conduct experiments to obtain the different parameters that we need for different tissues. This can then inform the model and therefore inform future experiments for different types of tissues. And then finally, something which would be much further down the line, we could then consider a kind of freezing protocol within this tissue and within this model. And we would introduce an equation here for temperature after we have reached CPA equilibration. And we can take a look at what happens to different substances as they change as the temperature changes also. So to finish, I would just like to acknowledge my funding bodies, and I would just like to say thank you to my collaborators and to my supervisor, Dr. Benson. And of course, I am happy to take any questions. Thank you.